Continuing with the report. It's going to be several videos long. I apologize, but we only have 15 minutes for a video each. So
31. Alexander S. Jones concluded, "We must seriously consider a laboratory origin for this virus, because 5% of both these influenza ARNA sequences share no known homology in any public databases. Blast sequence homology of swine flu indicates both the hemagglutinin surface protein as well as the non-structural NS1 interferon inhibition proteins are novel recombinants previously unidentified in nature. Both these influenza proteins, based on the genetic sequences released Friday, May 1st by the U.S. Centers of Disease Control (CDC), share their closest genetic identity with turkey, avian, and pig, swine, strains from multiple continents including North America as well as Asia. Even the closest matches indicate 5% previously unidentified genetic material. I submit this evidence, coupled with the lack of the presence of this virus at the pig farm near the proposed CDC's Patient Zero, a 5-year-old from La Gloria, 80 kilometers away from the pig farm in Parod, Mexico, shows that the origin of the flu outbreak remains unidentified at this time, and cannot be ascribed to Mexican or North American swine. Furthermore, I submit that since 5% of both these influenza ARNA sequences share no known homology in any public databases, in addition to the avian-swine hybrid nature of both these critical genes, that we must seriously consider a laboratory origin for this virus. Future research that may be promising includes identifying critical SNPs, especially in the PB2 and the NS1 coding regions, which may be markers for evolution of pathogen virulence, and should be closely monitored. The hemagglutinin protein should also be monitored for acquisition of a polybasic amino acid site, which would give the virus pantrophic properties as in the 1918 pandemic. Alexander S. Jones. 7. Evidence as to the role of Baxter and WHO in producing and releasing pandemic virus material in Austria. Baxter Pharmaceutical http://www.baxter.com/ has been chosen by the WHO to lead the efforts in finding a vaccine cure for the swine flu H1N1 virus. Baxter AG, headquartered in Vienna, and the Austrian subsidiary of the pharmaceutical company Baxter International, headquartered in Deerfield, Eel, USA, sent vaccine material contaminated with deadly live H5N1 bird flu virus to 16 laboratories in 4 countries in winter 2009 before a technician caught the mistake. The deadly mixture of live bird flu virus and human flu virus were mixed in a biosecurity level 3 facility, where basic protocol and procedures would make it impossible to ever mix a live virus bioweapon with vaccine material by accident. In the first place, the strain of bird flu that is lethal to humans has no place in the Baxter facility in Austria. So what was it doing in the facility, designed for research into normal flu viruses and vaccines and their production to begin with?
did you catch all that?
32. The material released was a combination H3N2 seasonal flu viruses and live, and labeled H5N1 viruses. If both strains were to incubate and recombine in a human host, a virulent airborne weapon that would cause a pandemic would be released, potentially killing billions. The Baxter facility did not radiate the material before they sent it out, leaving the deadly virus alive. To sum up, Baxter International, a global pharmaceutical corporation that has secured lucrative contracts to supply bird flu vaccines in pandemic, mixed live bird flu with human flu vaccine material in a biosecurity level -3 facility, failed to radiate it and sent it under a false label to 16 labs as vaccine material. About 8 weeks later, a artificial, genetic worldwide interspecies flu pandemic breaks out in Mexico City, close to another Baxter facility, and the same company is given government and WHO contracts to produce vaccines for the outbreak. According to Austrian Health Minister Alois Stoger, 72 kilograms of vaccine material was contaminated with the live bird flu virus which WHO supplied. http://www.parliament.gv.at/exev/ab_14575/f-name-oregon_1588554.html Parliamentary th Answers 1457 ab 24 gp May 20th 2009 Froggen 14 and 15 Das Verforschungswerk Bestimtes Material minus 72 kilograms waren als kontaminiert anzusehen, Word. In die Firma zurückgeholt und kontrolliert vernichtet. It is still not clear how 72 kilograms of the world's deadliest bioweapon can be sent by accident, from a high biosecurity facilities, not irradiated and under a false label. However, we know from Baxter itself that it produced the 72 kilograms contaminated material, using a wild-type live bird flu virus obtained from the WHO Reference Center. http://www.promedmail.org/pls/oat/f P equals 2400, 1001 to 53103, no, F2400_P1001_back_page, F2/400_P1001_pub_mail_ID, 10001, 76322. A statement on behalf of Baxter. I would like to provide the following update to a posting on ProMed, dated the 25th of February 2009. Avian influenza, accidental distribution, Czech, Rep, X Austria, RFI. The H5N1 strain was the A, Vietnam, 1203-2004 strain, received from a WHO Reference Center. All information concerning this incident has been provided to the involved national authorities and appropriate international bodies such as ECDC and WHO. Christopher Bona, Director, Global Bioscience Communications.
Sorry about that. But it's like I said, I keep having problems with other applications opening up without being prompted.
33. Corporate Communications. Baxter International Inc. 1 Baxter Parkway, Deerfield, EL 60015. Christopher_Bona at Baxter.com. Also, Baxter is the only flu vaccine manufacturer to work with wild-type flu viruses, felt to be more dangerous than the altered and attenuated, weakened, viruses other manufacturers use. http://channel_health_news_details.asp news_it=27,436 and news_channel_it=102 and channel_it=1020. The Austrian police have launched an investigation into the incident that almost triggered a global pandemic. The mixture of the deadly H5N1 virus with a mix of H3N2 seasonal flu viruses is classified as one of the most deadly bioweapons in the world with a mortality rate of 63%. So, with the Baxter incident in Austria, there is proof that Baxter not only created flu material with help from WHO, but also distributed them in large quantities to trigger a pandemic, while also positioning themselves to produce the vaccine allegedly to protect against the virus they created and released. In criminal charges filed against Baxter on April 8, 2009 at the Vienna City Prosecutor's Office. Landesgerichts TR11, 1080 Vienna, Austria, it was alleged that Baxter unlawfully, willfully and knowingly, in the period between December 2008 and February 2009, employed manipulative and deceptive devices and contrivances in violation of national and international laws on the manufacturing, possession, release and dissemination of biological weapons of mass destruction, and on organized crime, to manufacture and distribute a biological agent that is classified as a bioweapon among the population in order to profit from the pandemic. First, Baxter manufactured influenza material contaminated with a bird flu virus in its biomedical research laboratories in North on the Danube in December 2008. Baxter uses BSL-3, Biosafety Level 3, precautions in its laboratories, a system for the safe handling of toxic substances, which makes an accidental contamination of ordinary flu material with the dangerous bird flu virus virtually impossible. The 72 kilograms of contaminated vaccine material contained a mixture of a seasonal H3N2 human influenza virus and the deadly bird flu H5N1 virus. By adding a virus of the type H5N1 to an ordinary flu virus of the type H3N2, the H5N1 virus is restricted in its human-to-human transmissibility, especially because it is less airborne. However, when it is combined with seasonal flu viruses, which are airborne and easily spread, a new bioweapon is created. Second, Baxter distributed via this contaminated vaccines using false concealment and a false label to 16 laboratories in Austria and in other countries at the end of January, beginning of February, potentially infecting at least 37 laboratory staff, who had to be treated preventively for bird flu and ordinary flu in hospital. A total of 18 laboratory staff belonging to Avir had to undergo preventative treatment for the bird flu and ordinary flu at the Otto Wagner Hospital in Vienna on February 9, 2009, because of their exposure as part of their work to the highly pathogenic bird flu virus. 34. This indicates that, in the opinion of medical experts, there was a risk that the staff of Avir had contracted bird flu, and, unknowingly, acted as carriers of a pandemic virus into the population of a densely built-up Vienna city district, and in wintertime. The material was only discovered when staff working for Biotest in Kanaravis in the Czech Republic tested the vaccination on ferrets, who then died. Biotest was supposed to test anti-flu vaccination that should serve Europeans during the next flu season, and the labels on the material sent to them from Baxter via Avir gave no indication of the lethal contents. The 13 Biotest staff were treated with Tamiflu and were placed in quarantine for fear they had been contaminated with the bird flu virus, which is on the list of the possible biological weapons, and one of the most dangerous biological agents on the earth with more than 60% death rate. Subsequently, the same problem of Baxter contaminated vaccine material was found in the laboratories in Slovenia, Austria and Germany, who had received the material from Baxter. First, the company Baxter evoked the trade secret and refused to explain how a level 3 biological warfare pathogen found its way into H3N2 material, regardless whether or not this experimental vaccine material was intended for eventual use in humans or not. Baxter representatives have said that the material sent to the Czech Republic, Austria, Slovenia and Germany was in fact a pure H5N1 sent by accident, maybe to mask the previous assumption that it was in fact an ordinary flu vaccine, which was contaminated. It is still not clear whether it was in fact the pure H5N1 or contaminated vaccine. The Austrian Health Minister Alois Stoger confirmed on May 20, 2009 that the 72 kilograms of contaminated vaccine material has been destroyed, but no information has been released as to the genetic sequences of the contaminated material or what clade was Baxter's H5N1 vaccine from, whether from clade 1. Clade 2. Clade 3. Other. Therefore, it is not possible to know whether H5N1 resembles the strains circulating in waterfowl. Was the contaminated H5N1 strain genetically engineered? If so, by whom? Does the NS protein in Baxter's H5N1 material contain polymorphisms which suppress human interferon production? Was Baxter's H5N1 a full set of influenza genes, or was